Hello, my name is Drayton Standridge, and today we're going to be going over glucometer validation and measurement of glucose levels in cell media after hypoxia treatment. For this video, we're using the Hypoxia glucometer, as well as the iHealth glucose test strips, as can be seen here. Now we will be discussing how to validate the glucometer in order to determine its capabilities for media measurement. To validate the glucometer, we used DMIM media and diluted it in PBS with a dilution factor of 80%, so that each new dilution has 80% of the media the tube before it had. We used centrifuge tubes and diluted using 96 microliters of media and 24 microliters of PBS. We diluted 13 times to ensure that the glucometer gave us the words low, ensuring that we could not dilute any further. Here, we're demonstrating one of our dilutions as we move 96 microliters of one of our concentrations to the next concentration in the next centrifuge tube. Now, we are placing our dilutions on parafilm in order to test the glucose readings. We place 3 5 microliter drops per dilution and test them using the glucometer, and then we calculate the percent differences in the glucose readings. This allows us to validate our glucometer. It should be noted that you should also test your pure media in order to ensure that your dilutions occurred correctly. Now, we will be taking the glucose measurement of our dilution drop using the glucometer. To turn on the glucometer, first place a glucose test strip into the glucometer, as can be seen here. Then, you should see a liquid droplet icon, and this means the glucometer is ready to use. Then, simply place the glucose test strip up against the media until it makes a beep and begins counting down. Once this happens, you just simply wait until the glucose levels show. To dispose of a glucose test strip, simply use the switch located on the back of the glucometer and push it down. This should eject the glucose test strip. Now, we will be testing glucose levels in the media after placing cells in a hypoxia chamber. To begin, you need to have already split your cells and counted them using a hemocytometer. The links on how to do both of these are in the description below. After counting, use your cell count to determine how many microliters are required for 1,000, 5,000, 10,000, and 20,000 cells. Then you need to make sure you sanitize and place 2 24 well plates, a 25 microliter serological pipette, a 15 microliter serological pipette, and the media into the hood. After splitting, place 1,000 microliters of your cells into a microcentrifuge tube. Now we're going to label our 24 well plates. First, label your control plate. When labeling your plate, write 1, 5, 10, and 20 in the same places as the video. Then write GLC in the same place as the video as well, and CTRL to show this is the control well plate. The 1, 5, 10, and 20 represent the 1,000, 5,000, 10,000, and 20,000 cell groups. The GLC is a glucose-only control group. Now, onto your hypoxia group. This group will be labeled similarly as the last one, except without the glucose control group, and will be labeled HYP for hypoxia. This is what your control well plate should look like, and this is what your hypoxia well plate should look like. Now, it's time to add your media to the wells. Your media should be added to all of the following wells being shown in the video. For your control well plate, it's important to use the 25 milliliter serological pipette. After filling your serological pipette with an adequate amount of media, place 1 milliliter of media in each of the shown well plates. When you're finished, your control well plate should look like this. Now, we're going to do the same thing with our hypoxia cells, except with a 15 milliliter serological pipette. When finished, your hypoxia group should look like this. Here's the control well plate one more time. Now we will be adding our cells to the well plates. You should add your cells based on the math mentioned previously. For example, if you calculated that you need 100 microliters of cells for the 10,000 cell group, then add 100 microliters of cells to each of your three 10,000 cell wells in both your control and hypoxia well plates. Please ensure not to add any cells to your glucose control group, as this should just be pure media. Now, we're going to use the glucometer to measure glucose levels. Use the glucometer to take 3 glucose measurements per well for each of your wells. When using the glucometer, follow the same procedures mentioned earlier. It is also important to mention that for this treatment, the best results are obtained by dipping the test strip just below the surface of the media. When you are finished, place the control well plate into a 37-degree incubator. Then, place your hypoxia well plate into a hypoxia chamber. Then, run your hypoxia treatment for 4 hours. For the protocol on running a hypoxia chamber, check the description below. After 4 hours, remove the well plate from the hypoxia chamber and the well plate from the incubator. Once again, take 3 glucose readings per well for all wells and place them both in the incubator. On the 3rd day of incubation, take your cells out of the incubator and take 3 more readings per cells once again. This concludes the hypoxia experiment. This has been glucometer validation and measurement of glucose levels in cell media after hypoxia treatment. For other cell procedures, please check out the CBEC YouTube account.